Hello, my name is Graham Hatfull. I'm a professor at the University of Pittsburgh and a Howard Hughes Medical Institute professor. And we're going to talk today about bacteriophages, their genes and their genomes. First of all, in part one, I'd like to just discuss what bacteriophages are, some of their biological properties, and how they were discovered. So bacteriophages are viruses that infect bacterial hosts. So just as we and many of our animal cousins are infected by viruses that are specific for us, bacteria also have viruses that infect them, and they're called bacteriophages, or phages for short. They were discovered or co-discovered by Felix Durrell and Frederick Twort in between 1915 and 1918. And they were discovered as agents that, when added or present in a culture of growing bacteria, were capable of killing the bacteria and essentially having a bactericidal effect. It was really Felix Durrell who developed an assay called the plaque test, where he was able to take samples of bacteriophages that were able to kill bacteria, and he made dilutions, increasingly greater, a series of dilutions of the bacteriophage sample, and then plate that out in the presence of the bacterial host on solid media using like Petri dishes as shown here. And what he saw was that when he diluted out the sample sufficiently, he could see individual areas of killing, as you can see here, with smaller and large areas where the cells are dead and where the viruses have grown. These are called plaques, and each one of these individual plaques has arisen by a single particle which was capable of infecting a cell, and as the bacteria grew across the surface of the agar dish, the virus propagated itself, multiplied until in the plaque here there may be perhaps 1 million or 10 million or more individual phage particles. This plaque test was really important because they could tell from looking at cultures of, or looking at lysates of bacteriophages, that there was nothing to see in there. They could filter the samples. They knew they still had the infectious property, but the tube looked completely clear. There was nothing to see. And when they placed these particles with this capability in the light microscope, there was nothing to be seen. So these were mysterious entities, but Felix Durrell showed conclusively that they really are particulate in nature. It was somewhat later, in the late 1930s and 1940s, that the electron microscope was developed, which has a level of resolution way beyond the light microscope, and was able to show for the very first time what viruses, including bacteriophages, actually look like. And I'll show you some pictures in a minute, but this just shows a stylized example of what one of those phages looked like. At the very top here you can see is a structure that is referred to as the head. Sometimes it's called the capsid. That is attached to this longer structure here, which is the tail. And the DNA, or the genetic information that the virus carries in order to multiply itself, the instructions for its own replication, are carried here in the virus head. And so we can see, and we now know of our understanding of what this particular virus and these types of viruses look like, is that this structure here at the very bottom, which is the tip of the tail, that is the region that recognizes and binds to the outside of the bacterial host. During the process of infection, the structures that we see, principally constructed or made up of protein components, that stuff stays on the outside of the cell, and the DNA, or the genetic information, is what passes from the head through the tail into the cell and then reprograms that cell in order to make more copies of the virus. By electron microscopy, we can see, and we now know, that a very large proportion of these naturally found viruses are in an order which are referred to as the Cordovirales, of which these are primarily phages that contain tails, as I just described and shown in these examples here, and they contain double-stranded DNA. So there's lots of different types of viruses in different shapes, but the vast majority that you find in nature fall into this particular order. And these have names according to the types of tails that they have. These are the Myoviridae, and they have contractile tails, and so the tail actually contracts like a syringe when the DNA is injected into the bacterial host. These are called the Podoviridae, which have little short stubby tails, and these ones on the right here are the Cyphiviridae, and these have long, non-contractile and flexible tails. So these are the main forms of these viruses. There are, however, a variety of number of different types of viruses that have different shapes and have indeed different types of DNA or RNA genomes within them. So we can think about the various steps that are involved in the propagation of a bacteriophage during this process known as lytic growth. The phage starts by adsorbing to the outside of the cell. The DNA is injected inside the cell in this process of penetration. There is a set of early proteins which are encoded by the phage, but which uses the host machinery to express them. Viral DNA is replicated to make lots more copies of the virus DNA. And then these late proteins are expressed that make the structures that I just showed you in the electron microscope. The capsid structure gets assembled, the tails get assembled, and then in a process of DNA packaging, the DNA is stuffed into those heads until the heads are full. The tails are attached, and at the very final step of this process, the bacterial cell is going to lyse with enzymes encoded by the phage genome to break open the outside of the cell, and the progeny viruses, typically 50 to 100 new progeny viruses will be released, and to go on and repeat this cycle whenever they find another bacterial host. So while many phages go through a lytic growth cycle, and that is essentially simply how they reproduce themselves, that's not the only type or form of growth cycle that phages can enjoy. And there's a large class, perhaps even a majority of bacteriophages, that enjoy what is referred to as a temperate life state. What that means is that when a temperate phage infects its bacterial host, there are two possible alternative outcomes. One of those outcomes is lytic growth, in which they propagate themselves in exactly the same way as I described to you in the previous slide. And normally that occurs perhaps 80 or 90% of the times that the host cell is infected by a temperate phage. 10 to 20% of the time, there's an alternative outcome. And that alternative outcome is referred to as lysogeny. In lysogeny, the lytic genes, the genes that are required to propagate itself and lyse and kill the cell, they're all switched off, they are repressed, and the phage DNA establishes itself so that it can be propagated in the long term within the subsequent growth cycles of the bacterial host. And that's usually, although not always, accomplished by integration of the phage DNA into the bacterial chromosome. So the phage genome itself becomes incorporated and becomes a part of the bacterial chromosome and gets replicated just like all of the other bacterial genes do. So we can think of lysogeny as a form of parasitism, where the phage is simply going along for the ride in what is otherwise a very healthy cell. it's a free ride through many, many generations of bacterial growth. So lysogens tend to be stable, but they are not they don't have to remain in that state forever. They can go through many, many, many rounds of growth. However, as a process that happens either spontaneously or can be induced by DNA damage, such as UV light, lysogens can leave the comfort of that life cycle, and they can be induced into full lytic growth. There's a relatively easy way to distinguish between phages that are temperate and those that are lytic and can only undergo the lytic growth cycle by the types of plaques that you see on agar plates. Lytic phages form simply just clear plaques, as shown in the bottom right-hand corner here, where all of the cells within that infected area have been killed by phage infection. Over on the left at the bottom here, you can see what temperate phages look like. They form turbid plaques. You see plaques because there's cells that are being killed through lytic growth and propagation of the virus. But there's that important and key subset of cells that in which lysogeny is established, and those lysogens are able to survive and to grow, and they can grow perfectly happy even though they're bathed within this density of bacteriophage particles.